Hello, my name is Sara Martinez. I am a PhD and student in biomedical sciences at ISCB-LA, and I am going to present my project entitled Reconstruction of the E. coli leaf pathogenicity island network. Enteropathogenic E. coli and enterohemorrhagic E. coli form a family of bacterial pathogens that bind intimately to the intestinal epithelium and destroy the microvilli, forming characteristic lesions called adhesion and erasion, causing acute diarrhea in humans. Despite medical importance, there are few efforts to build the regulatory network of the genes related to the island. It has not been introduced all the information to the complexity of global and local regulation. The regulatory network contains the genes belonging to the leaf island and some non-encoding genes regulated by leaf regulators in the PEC and EGEN strain. Our network comprises 239 regulatory interactions obtained from 250 papers, including 43 transcription factors and 84 target genes. Regulatory networks allow visualizing how biological systems are regulated, and they are being applied in several fields of knowledge such as medicine and biology, emergent properties, or those that cannot be observed and analyzed each node behavior alone. Network can have feedback loops with positive or negative regulation that affect the synthesis rate, some components of the network. Because of this, our primary goal is to introduce all known information about the island leaf regulation using assisted curation. Network interactions are classified as either direct or indirect depending on the experiment methods used to obtain them. The integers allows to find new feedback loops. The methods of this project are the following. First, it was an article selection. We built a data set of 250 articles through an advanced search in PubMed. Then we recovery of regulatory interaction from the lectures in the semi-automatic extraction of regulatory interaction was done using the ontogene system through the Odin interface. After that, we visualizing of the regulatory network of the leaf island using Cytoscape. Finally, we made a functional enrichment using Panther software. In addition to the extraction of regulatory interaction used ontogene pickle stack in the national information subject condition experimental method reference and some cases in the regulatory interaction is direct or indirect. Some examples are the table 1. In addition, we found other levels of regulations for each regulatory interaction such as post-transcriptional regulations or regulation by small RNAs. The figure represents the connection of the pathogenicity island leaf 1 to leaf 7. The shape and color of the edge represent the regulatory interaction, for which red arrow means activation. The green bar is repressions, and the blue node is an undefined regulatory effect. The shape and color code will be used an edge of the representations of the leaf 1 network. The hub nodes of this network are represented as circles. The type of the line represents the type of interaction of each regulatory interaction. The solid, dotted, and zigzag lines represent the direct, indirect, or known mechanisms respectively. The black and gray colors in the different shapes represent the strong and weak evidence respectively, and yellow color represents that is both weak and strong evidence. The conclusions are that assisted curation through Odin clearly showed to be an efficient tool to curate that in impact and or network contain 85 regulatory interaction of which the representative and dual nodes are learned, and GRLRA, the master regulatory of the pathogenicity line, and gene ontology analysis choose 3 of the main mechanisms used by EPEC and HEC to develop pathogenesis in the intestine. Input and output connectivities were calculated for each node network. Ka was defined as the number of the input edge of the node. Thank you very much for your attention.